Our next speaker is Chantal Hamel. She's at the Swift Current Research Station, originally from Quebec City. And she's going to be talking on mycorrhizae and diversity of fungi and some crop management, prairie management, cropping systems, or management of prairie cropping systems and different aspects of that. So if you can welcome Chantal to the mic, please.
Thank you very much. Yes, so it's a pleasure to be here today and to have the opportunity to talk to you about some of the latest results that we obtained on the mycorrhizal symbiosis, in particular in relation with nutrient efficiency of cropping system in the prairie. Just a point to say that we think that it's important to improve the nutrient efficiency of cropping system, because inefficient use of nutrient is not only a waste, but also it's a cause of pollution, which has a lot of impact on the sustainability of the planet, actually. So we need to change the way we think about things, and one thing that's important to realize is that nutrient management, a lot of it is done by soil microorganisms. And in the past, we didn't have much handle on that, but more and more we do. We have new tools. And we know microorganisms are doing lots of work in the soil, but they don't work for us. Sometimes, they work against us, especially for nutrient use efficiency. I'm going to talk to you mostly of the mycorrhizal symbiosis, especially in wheat-based cropping system. I will talk about the diversity of the mycorrhizal fungi in crop fields, the impact of these fungi, and ways we can manage them. And I would like to talk also a little bit about the possibility of breeding wheat for improved mycorrhizal symbiosis. The thing with wheat is such an important crop in Western Canada, but it does not respond very well to the mycorrhizal symbiosis, and mycorrhizal fungi, they are everywhere. So we know the mycorrhizal fungi, well, they are part of the root system in most crops. They facilitate nutrient uptake by carrying nutrients in their hyphae, which act as pipelines. However, this ecosystem service is not free, and the plant have to provide carbon from photosynthesis to the fungi for the system to work. And this is a trade-off that may explain the results we observe in a survey we conducted in the prairie. Here we are looking at some data that we collected from 72 organic wheat fields. And we were surprised to see, actually we were looking at what are the drivers of wheat productivity and efficiency of nitrogen uptake, phosphorus uptake. And what we found is that a group of mycorrhizal fungi that is not well classified yet, so I call it here Glomerinaceae insecticidis. It's a group of mycorrhizal fungi that was negatively related with wheat productivity, with nitrogen, efficiency of nitrogen uptake and phosphorus uptake. That is, when we had more of these fungi, we had less yield, we had less nitrogen and phosphorus in the plant per unit of nitrogen and phosphorus in the soil. And this was unexpected, of course. In contrast, I show here in green, under efficiency of phosphorus uptake, that the abundance of other mycorrhizal fungi belonging to the group of Claroideoglomus and Diversisporaceae were positively related with efficient phosphorus uptake as we expected. We didn't see the same negative relationship with the group of not well-defined mycorrhizal fungi in conventional wheat production. Maybe for one thing, that these fungi were making up a lower proportion of the mycorrhizal fungi in conventional wheat fields. We had surveyed 78 conventional wheat fields throughout the prairie. What's interesting to see here is that, again, we see positive relationship with efficiency of phosphorus uptake and nitrogen uptake with mycorrhizal fungi of the Rhizophagus group and the Claroideoglomus group. Maybe two things to remember here is that mycorrhizal fungi and other soil microorganisms, they do contribute to crop production in conventional system. It's not because we're using fertilizer and pesticide that these organisms are inactive. They do contribute to productivity in good ways and bad ways, whether or not we think about it. Another thing is that it's important to understand that the mycorrhizal fungi are not all the same. Mycorrhizal fungi make up a big group of fungi, and here I'm showing all the lines, are different strains of mycorrhizal fungi that we found in the prairie, and in the red circle is the proportion of the fungi that they are not beneficial when we blow it, that circle. So we can see several strains that are associated with Indicum and Irenicum that are not seemingly very good mutualists, at least with wheat. And these fungi, they are well diversified in prairie soils and sometimes abundant. As we can see here, the scale when it is more like tan color, it's more abundant. When it is purple, it has low abundance. So we can see that some of these not so good mycorrhizal fungi can be quite abundant, especially this Glomus indicum number 86. So it's not because we have negative relationship between a group of fungus and wheat yield that these fungus are causing necessarily reduction in wheat yield. Because, for example, these fungi may be liking dry soil, and as we know, wheat yield doesn't like dry soil. We have less yield in dry soil. So it could be something like that. But other information from another study that we conducted at Spark, in Swift Current, at the South Farm, we could see the same negative relationship in chickpea yield. We had a trial with lots of different chickpea genotype. And it was all grown on the same piece of land at the South Farm. And we could see, again, that when we had less or maybe more of these fungi that are suspect to be not beneficial, we could not have high yield. So we need to work more on this, but we think that we could have a problem with these mycorrhizal fungi, this group. And to resolve the problem, maybe in fields where we have lots of these fungi, these species, we could use the inoculum that is available for sale everywhere in the prairie since 2010. This product is MyPro, and it contains a strain of Rhizophagus irregularis, actually. On the label it says Glomus intraradices, but the name has been changed to Rhizophagus irregularis. And it's important to remember for the data I'm going to show here. So this data is the diversity of the mycorrhizal fungi in the root of wheat. So the upper panel is the root of wheat. I don't know if I can get that, no, anyway. So the 4 bars on top are showing what's in the root of wheat, and the 4 bars of the bottom are showing what is the diversity in the soil around the roots of wheat, the same roots. And we show the effect of different previous crop. But what is most interesting here is to see that what's in the root doesn't correspond to what's in the soil. In fact, wheat will select different species from the soil and form the association with these species. And the species that we like is species of the Rhizophagus group. So it means that we have a way to to manage the mycorrhizal fungi in the soil if we can, select the plants that will do well with the good species. We didn't do much work with MicPro, but we had one field experiment, and I'm showing the result of this experiment here on the root colonization. So we had 4 different wheat cultivars, and we inoculated these cultivars or not. And we can see here that in 3 of 4 cultivar, inoculation increased the amount of root colonization at the 5-leaf stage of wheat. But it was true for 3 varieties, but not in Pellissier, again showing that the plant can affect the outcome of the symbiosis, and it's something we can work with. We didn't see yield increase in this experiment with inoculation with MicPro, but why we didn't see a yield increase? Maybe because we didn't have much of these fungi that we think may have a negative impact on wheat. As you can see here in red, we had only 1 strain of the negative, the not so beneficial group, and it was making up only about 1.4% of the total mycorrhizal community in the soil. This result is from Cuba. In Cuba, they put a lot of resource into developing inoculum of mycorrhizal fungi, and they're pretty good at it. And they developed different inoculants. They made tests, lots of tests, and here is the result of some tests where they used the different highly effective strain in different soils on different crops. So the different crops are different forage grasses. And the thing that this graph shows is that the highly effective mycorrhizal fungi will work well only if we put them in a soil environment where they can be effective and produce their effect. And what we can show is that, for example, the Funeliformis mossae was highly effective in the clay soil, whereas in the nitty soil, the Glomus cubens was most effective, and in the Camby soil, the Glomus species was most effective. So this is another thing that is going to help us develop inoculants that are highly reliable. And I'm sorry for this slide here, but I wanted to show you the last results we obtained from a test similar to what I have shown, where we tested different strains of mycorrhizal fungi. They were indigenous of the prairie soil, and we tested them in different crops and different soil type. And we can see, particularly in the lentil here, that they have different effect on the lentil and other crop too, but lentil is more strong effect in different soil. In black soil, the effect was very poor. We had growth reduction, as shown by bar under the 0. We had better result in the brown soil. In the dark brown, we had good result with the fungus called irregularis, not so much with the mossae. And in the dark gray soil, we had, some irregularis and some mossae that were more effective. So this is very promising for bringing this technology in a better format to prairie growers. One question that I had in, driving around in the prairie and seeing how intensive is agriculture. In some areas, the only thing you see is crop as far as the eye can go. And this mycorrhizal fungi, they are obligate biotroph, means they cannot live without a plant. And especially in the brown soil zone, where we even have some year of fallow, maybe we are losing diversity. So in our study, we went around the field, and at the same time, we sampled the roadsides, and we sampled also some natural area we could find. So in the dry area, it would look like the photo at the left, and in the parkland, the photo at the right. And what we found is kind of good news, is that the mycorrhizal fungi are pretty resilient to crop production. And we found only one species that was only present in the natural area. And most species were present under all type of land use, and especially the common species were found everywhere, just that they were found in different relative abundance, and that's what is shown in the graph in the right-hand side, where we can see, for example, the bottom number 119, Glomus iranicum, look like it likes very much natural environment where it proliferates pretty good. In contrast, number 9 on top looks that it's liking more the roadside where it grows better than in other area. Here, more effect of land use type. We add, for comparison, 20 sites from Nova Scotia where they have very different soil, very different climate. And what we find is that in red, the red dots are the field in the prairie. This is the circle. And field in Nova Scotia, the triangle, so the red are under wheat production. We can see that they are similar. So that shows that cropping actually has an influence that is similar in the two areas. In blue, we have the diversity, the structure of the mycorrhizal fungi in the roadside, and we can see that it can be quite different. The little graph on the right is quite interesting. It shows species richness, that is the number of different species of mycorrhizal fungi in any average sample of roadside or cropland or natural area. And we can see that the roadside really hold a lot of diversity. So it is good news, because if we are losing diversity in the field, we can always recover it from the ditch. So in conclusion, for that section, we have seen that we probably have good and bad mycorrhizal species in our fields. MyPro, the inoculant that is commercially available, may outcompete the bad mycorrhizal species for wheat occupation, because it seems that the rhizophagus are quite competitive. Soil type is a determinant of plant response to mycorrhizal fungi, and this is something we have to work on more to provide inoculant that work more reliably in the different soils. And we found also good news. The mycorrhizal fungi are quite resilient to human disturbance. And despite that we have some shaping of the diversity of mycorrhizal fungi due to agriculture, we have a good place where we can go back and find these species that is in the roadsides. Now I would like to talk to you a little bit about our work to show if we can improve the mycorrhizal symbiosis of wheat, and not only because wheat doesn't respond very well to the symbiosis and mycorrhizal fungi are everywhere, but also because wheat is such an important crop that if we can improve the quality of the soil microorganism by having wheat that are creating good soil biology, then that would be good for the whole cropping system. So actually I'm going to talk to you about this greenhouse experiment we conducted at Swift Current. We had three main questions when we ran this experiment. First, is there enough genetic variation so we can select wheat genotype using conventional breeding technique? We have a big breeding program for wheat at our research center, and they use traditional breeding technique. Second question was the ability of forming effective symbiosis lost in modern wheat cultivars. You know, wheat is a crop that was very much improved, and it is improved under condition of high soil fertility. So some people have proposed that this condition of selection may have weed out the genotype that are able to form effective symbiosis with wheat. So that was an important question we were asking. And lastly, we were interested to find a trait that would allow us to select rapidly the promising genotype among the large number of plants that needs to be looked at when you are selecting genotypes. So the experiment was, as it's shown here, we use a lot of different genotypes. At the bottom, you have the group of the genotype that were introduced in North America at the beginning of the last century. And we compared these to what we call modern genotypes, those that are produced in Canada or North America after the event of nitrogen fertilizer after World War II. And we have a special class on top that are the varieties that were produced, that were released in the last 10 years. So what we found is that we have variability throughout the groups, so it is possible to select for better genotypes. We found also that root colonization, looking at root directly, is not a good indicator, it's not a good trait to look for growth response. This is shown by the 90-degree angle between growth response and root colonization. However, it was interesting to see that growth response was negatively related with the response in terms of nitrogen concentration and potassium concentration in the plant. And this indicates that, okay, the higher concentration of nitrogen and potassium in the leaf, the lower the biomass of the plant. So this indicates that the plant is actually limited by carbon. And this, corresponds to what we've heard this morning with Dr. Martin Perry. So crop can be carbon limited, and it looks like it is the case in certain wheat that cannot fix enough carbon for their own need and for the support of the mycorrhizal fungi. So we did a little more maths, and here we are looking at the relationship, a positive relationship between the response of wheat variety to mycorrhizal inoculation and leaf nitrogen and potassium concentration. And here it is the inherent concentration in the leaf, that is when the plants are not mycorrhizal. And we can see that the higher the nitrogen and potassium, the better the response. And this is explained by the fact that, of course, nitrogen and potassium are very important nutrients for photosynthesis. So we propose here that leaf nitrogen and potassium can be used as indicator for efficient mycorrhizal symbiosis in wheat. So in conclusion from this section, we've seen that crop plant can be bred for better symbiosis. Leaf nitrogen and potassium level can be used as a trait, as indicator of symbiotic effectiveness in wheat. And the fact that symbiotic effectiveness was not lost in the modern variety is a very good news, because it means that wheat breeders do not have to go back in genotypes that they don't know. They can use the material that they are using commonly to develop new variety to screen out some genotype that will be effective at making good mycorrhizal symbiosis. So this is all I had to present to you today. I'm not sure if we have time for questions, so I would be happy to answer any question you may have. Yes?
You were saying that these need carbon, so has there been any work done when you have manured soils? Is there a higher level of these fungi that are beneficial for wheat?
Yeah, that's a very interesting question. The sad thing is that these fungi, as far as we know, they are not able to use carbon from other sources than the plant. However, what we've observed is that soil organic matter somehow increase their abundance. So maybe we didn't figure out yet how they can use the carbon in the soil, but yeah, we find them more abundant, but for now we cannot say that they are using the carbon from the soil because we have never seen it.
Don't think Rick Talliu's around. He won't like this one. How far can you throw, Rick? Anyways, brassicas, such as canola, are generally, they're considered a very poor host for mycorrhizal activity. And I was looking quick at the label on this Mica Pro. It won't work in brassica crops. So how is the high levels of canola, and well, especially canola, but mustards as well, that we're growing in Western Canada affecting these levels? And how can we keep, or is it going to be possible to keep these levels of beneficial mycorrhizae high while we keep growing and pushing our canola rotations? So what rotations and cultural practices can we use to keep these levels high?
Yeah, very, very good question again. I'll try to make a simple answer. Yes, because it's connected with the previous question. You know, what we think is that the mycorrhizal fungi depend on the plant for carbon, but we've seen actually that in the black soil zone, where is the most intensive canola production, this is where we have the highest amount of mycorrhizal fungi. So maybe we just need to scratch more into that, and maybe the mycorrhizal fungi can somehow use soil carbon, or maybe they can be very long, persisting very long in the soil, because again, we've seen that cropping, you know, canola and also the summer fallow. We have lots of summer fallow in the southern Alberta, for sure. In Saskatchewan, it was like tradition to have a rotation wheat fallow, you know. So for fungi that depend on plant, that's not pretty good. But what we find is that, yes, they are there and they are pretty resilient to our cropping system. So maybe we just didn't find a way to see that they are using soil carbon. Or maybe, again, they're just able to stay in the soil for a long time. And know how to keep the mycorrhizal fungi to good levels with growing canola. Well, you know, when you're growing a crop, as everybody say, people want to grow money, actually, not fungi. So we have to do with the best we can with what we have. And if we want to grow canola, and we've seen, you know, research has shown that sometime after canola, if we have a crop that is highly dependent on mycorrhizal fungi, we may have yield reduction. So we may just use inoculum, or we may decide to grow wheat, for example, which is a crop that doesn't depend very heavily on the symbiosis. So the canola doesn't impact the next crop, you know, like canola, wheat, pea, or lentils. So that would be something we could do. Okay. No more questions, so thank you very much.
Thank you. Here, Oh, yeah, sure. We have time for one more.
Does spraying fungicides have any effect on the field?
Another very interesting question. We have been working also on fungicide. And what we find, interestingly, is that the effect of fungicide is not direct, because the fungi are protected in the soil. Same way, you know, it's very difficult to get rid of soil pathogens, soil-borne pathogen, because how can we get the fungicide down there? It's not easy. So the fungi are protected, but what happens is the plants, you know, plants, they are very interesting organisms. They don't walk. When there's a danger, they don't go away. They stay there, and they defend themselves. They produce chemicals that will inhibit pathogens to different degree. So when we protect the plant using fungicides, say for leaf pathogen, we did that work on chickpea. So lots of fungicide goes on chickpea. So when we put fungicide on chickpea, we have less disease in the leaf, but we have more in the roots because the plant doesn't defend itself against the root pathogens. So, but, you know, it's just a very interesting situation. That I don't mean that we should not be using pesticide, because if we don't use pesticide, for example, on chickpea, we don't have a crop.